So far in the course, the first half concentrates on the structure of the microbes, right? We did a lot of staining. The second half concentrates more on how they function, right? So these tests that we ran today and the results that we looked at, right, really look at what they can do, what's their functionality. Those things combined are almost like anatomy and physiology, right? As we do with multicellular organisms, we're doing the same with our little microbes. And of course that'll help us in identifying them, right? So structure are things like, does it have a capsule, right? That's a structure, right? And we did a stain to determine that. But questions like function or can they do something? And so one of the major questions we asked a lot with the tests that we ran today is, can they ferment glucose? Can they take that sugar, right, and get energy from it? So most of our tests, right, all the tests that we're looking at, right, are going to answer those types of questions, right? What can they do? So they're all classified as what we call differential, right? They either can or can't do these things, right? You got positive and negative results for some of them, and then there's a multitude of different results that we could detect in the Phenol Red test. But it's all about what can they do, right? What can they do? So redox is oxidation and reduction. It's basically the movement of what? Anyone know? Electrons, right? Oxidation, reduction, most people know the saying oil rig, right? Oxidation is the loss of electrons. Reduction is the gain of electrons. And that's what happens in chemical reactions. Somebody's losing electrons, someone else is picking them up. So redox is both reduction and oxidation, right? It's an abbreviation. So electrons don't just disappear, right? They've got to go somewhere. So they go from one molecule to another. In biological systems, they're usually accompanied by protons, and actually hydrogen atoms are just a proton and an electron. So when the electron moves, the proton moves. So a lot of times the whole hydrogen atom moves, right? But the important thing here is that movement of the electron. So lots of times, right, we see these plus signs, right? This is indicating that it lost electrons, right? It's positively charged. Over here, it gained those electrons and the hydrogen. So notice another hydrogen has been added. And an additional proton even came along. So this guy lost electrons, this guy gained electrons. And this is one of the major steps in the Krebs cycle. Did you guys remember those pathways that I mentioned last time for breaking down sugars? Organisms that respire aerobically. What's the first step for breaking down a sugar like glucose? Glycolysis. Then what's next? It's a cycle. Krebs cycle or TCA cycle, right? And then those electrons that we're taking off molecules, right, go to the Electron Transport Chain, right? And that's how we generate energy, right? So glycolysis is going to break down glucose into pyruvate. The good news is this is stuff you should have remembered from biology class, right? And it's not stuff we're not going to test you on. Notice I said review. Let's review. But it's pertinent to what we're doing and looking at, right? So pyruvic acid and pyruvate, they're basically interchangeable. Although chemically they're a little bit different. The next is converting that pyruvate with acetyl-CoA, right, to go into the Krebs cycle, right? So there's a little step right before it goes right into the Krebs cycle. Krebs cycle is also sometimes referred to as a tricarboxylic acid cycle, or abbreviated TCA. Another name sometimes you'll see is a citric acid cycle, because citric acid is formed, right, or citrate is formed in the process of the Krebs cycle. And then we have the electron transport chain. So electrons that are picked up by electron carriers from glycolysis, from the gateway step, from the Krebs cycle, can go to the electron transport chain And if we're talking about an aerobic organism, what's going to accept those electrons? An aerobic organism. Oxygen, right? And it forms water. So ultimately, carbohydrates, like glucose, are oxidized. We remove electrons from them. We're going to release carbon dioxide in that process. Right? We're also going to take any free energy and store it in the form of ATP that can be used later. As I said, reduction and oxidation happen together. Right? If someone loses electrons, someone else gains them. Ultimately, in the case of the gaining of electrons for aerobic organisms, I said it's oxygen. And when it forms it again, it takes some protons, so then we have water that forms. So here's good old glucose. It's a 6-carbon sugar. Pretty little sugar. There's different pathways of glycolysis. The most commonly studied is the Edmund Meyerhoff pathway. He, I guess, was the third guy, too. Most of the time he gets forgotten about. Right? So obviously, scientists that were involved in that. It's called a carbohydrate because its chemical formula is every carbon you have a molecule of water. Right? So that's why sugars are sometimes referred to as carbohydrates. Usually when you're looking at your nutritional labels, they're talking about more than one sugar. Carbohydrates are usually talking about a whole bunch of sugars hooked together like in the form of starch. When they're talking about simple sugars, like 2 sugars together, like sucrose or glucose by itself, that's what's on your nutritional labels as sugars. Right? So those can be quickly broken down. The individual sugar components don't have to be broken apart first. So this is why, like, if you chew a cracker, crackers contain a lot of starch, it starts tasting sweet over time because you start digesting and breaking apart the sugars. And you can actually taste them. So in the process, I said we're going to remove electrons. And the electron carriers like NAD pick up the electrons and protons. And that's what's happening here. And even in this step, they can phosphorylate ADP to ATP. So even some ATP is produced in glycolysis. A little bit. Not a lot, but a little bit. So we call that substrate level. At the enzymatic level, this is happening. These electron carriers here are going to go travel to the electron transport chain for them to be converted into ATP, stored energy. So gateway step, again, notice more electron carriers, right, and even some carbon dioxide produced before we even enter into the Krebs cycle. So an acetyl group is two carbons. So first step of the Krebs cycle, see, we form from oxalate and the acetyl-CoA, we form that citrate. Right? So again, another reason why it's sometimes called that. And another reason is that you have 3 carboxylic acid groups associated with citrate. So that's another reason why it's sometimes called the Tricarboxylic Acid Cycle. Right? Because that's what you're forming in this cycle. So steps in the cycle, again, here they have some energy. It happens to be in the form of GTP. It's very similar to ATP. Again, substrate level phosphorylation. But then we're taking more electrons away from these molecules, right? And in the form of electron carriers, NAD, and another new one, FAD. These are just electron carriers, right? They're picking up electrons and they're going to carry them to the electron transport chain. We're going to give off carbon dioxide, right? The carbons that we give off are released in the form of carbon dioxide. That's where the carbon goes. And some of the oxygen. So the citrate test, right? We looked at that today, right? So the citrate test detects organisms that can take up citrate from the media. Not whether they can do the Krebs cycle and make citrate. Whether they can take citrate from the media itself. Okay, so that's an important distinction. The citrate test also detects organisms that can metabolize ammonia, which is NH4 4+. So it's very similar to, once they take the citrate in, they are going to process it through enzymes that are used in the Krebs cycle, but they're going to go in the opposite direction, which is possible. Directionality is usually determined by how much you have of a substance, right? It will determine which way the reactions go. So for the electron transport chain, these are embedded in membranes, right? So let me, my membrane one is the next one. Getting ahead of myself. So NADH, right, has picked up electrons. It's going to donate those electrons to electron carriers. These are proteins embedded in the membrane. So these are the different names for the protein complexes. FADH also donates its electrons to the electron transport chain, but it does it at a different point. And so the difference there is when this is happening, when these electrons are flowing, they're pumping hydrogen ions, protons, across the membrane. For NADH, they'll pump 3 sets of protons across. For FADH, only 2. Because it doesn't enter at this step. Notice at the end of the chain, right, we're talking about aerobic respiration, so oxygen is going to accept those electrons and you get the formation of water. So it's in a membrane for us, it's the membranes within our mitochondria. Right, where this is happening, where the electron transport chain is happening, right? In the inner matrix, you get the citric acid cycle running, right? The electron carriers carry the electrons to the electron transport chain. They pump them across the membrane, right, into this space. And then the protons travel back. When they travel back through, they travel through a port, a protein port. There's also an enzyme. This is ATP synthase. So that energy, that flow of the protons going back through is what allows the ADP to be turned into ATP, that stored energy in that phosphate bond. Now for bacteria, this will happen on their membrane itself, their plasma membrane. Okay? So that proton gradient, those hydrogen ions, is what generates the energy for making ATP. This we call oxidative phosphorylation, again, because electrons were used to phosphorylate that ATP, to add that phosphate. The problem with the use of an electron transport chain by an aerobic organism, one that's using oxygen, as you see here, is the final electron acceptor. Sometimes oxygen electrons will leave early in the chain, and the oxygen will pick up the electrons, and you'll form superoxide radicals. These are highly charged forms of oxygen. They're extremely damaging because they react with other molecules and interfere with metabolic processes. So if you're using this electron transport chain in the presence of oxygen, you're going to get the formation of superoxide. That's toxic, right? So we have to have some way to neutralize it. So your cells have enzymes that will neutralize this toxic form. The other toxic form of oxygen that can form is when hydrogen, oxygen, and electrons are donated together, and they can form hydrogen peroxide, which is what we use today, right? To run a test to see if they have the enzymes to get rid of hydrogen peroxide, right? Can we break that down and make it safe again? And the answer for all your organisms was yes, suffocate. Right? And there's a reason for that. So catalase test detects, right, the catalase enzyme. That's the enzyme that's going to break down this hydrogen peroxide and convert it into a safe form of water and gaseous oxygen. So that's why it bubbles, right? Because oxygen is in the gas phase, right? And so that's oxygen bubbles that are forming. So this formula is important for your report, right? Yes? No? Yeah. Okay. And then that's just balancing the equation at the bottom. But for every 2 molecules of hydrogen peroxide, you're going to produce 2 water molecules and a gas molecule of oxygen. And it's important that, you know, we write this, that is, it is in the gas form. Although most of us, when we write O2, we think of the gas form. But O2 can be in liquid form. It can be dissolved in liquids. So when we look at the breakdown, right, the catabolism, the breakdown of carbohydrate like glucose in the presence of oxygen, we generate water, as we saw. We generate a lot of electron carriers, carbon dioxide, right, and some already at substrate level ATP and AGTP. Those electron carriers go to the electron transport chain, and 3 ATPs are produced, right, because we've had 3 sets of protons going across for each NADH, and about 2 for each FADH2. So therefore, theoretically, right, aerobic respiration for glycolysis, gateway step, Krebs cycle, and electron transport combined, for each molecule of glucose, you could get up to 38 ATPs. That's a lot of energy. In eukaryotes, the yield is only about 36 because of that transport that has to happen in the mitochondria. We expend some energy. Glycolysis happens outside the mitochondria, and then that movement inside to the mitochondria, we lose some energy. So, as I said, some of them are produced at substrate level, 4 of them. 34 of them are produced at the oxidative level. Now that's theoretical. Actual yield is more like 29 to 30 per glucose molecule. And these pathways and stuff are reviewed in your appendix of your lab manual. Right, so energy-wise, right, we can generate 2,000 kilojoules of energy. Each ATP molecule generates about 30 kilojoules of energy. So if you were to times that by 30, right, we're only really around, for 1 molecule of glucose, about, say, 1,000 kilojoules of actual energy. So these, if you like to see stuff moving, these are some video links. I think I already had them in the folder for today in Blackboard. One of them is kind of goofy. The guy's like singing. It's cute stuff. So if an organism lacks electron transport chain, right, or can't use it because maybe oxygen's not available, okay, we have this problem of the buildup of the coenzyme, right, that electron carrier. It's stuck with the electrons. It has no place to take them to. So it can't pick up anymore if it's already full, right? So we have to have some way of getting rid of these electrons so that they can keep breaking down glucose, at least by glycolysis, right? So we get some energy production. So what's the answer? It's going to hand it off to products of glycolysis, and it's going to form things like acids, alcohols, and gases. Collectively, this process is referred to as fermentation. And some organisms just don't have electron transport chain and do this all the time, right? Other organisms, if oxygen is lacking, right, like our facultative anaerobes, right, they'll ferment instead. The problem with that is we saw with the yields, right, a lot less energy when you can't use the electron transport chain. Right? That's the take-home message from this. So when it comes to fermentation, there's lots of choices, right? Lots of things that different organisms have the ability to do. So remember, from glycolysis, we get pyruvate. It can't really go into the Krebs cycle because we're going to generate a whole bunch more, right, electron carriers that have no place to go. No electron transport chain. So we can change them into different forms. So formaldehyde is one of the things that forms when you remove carbon dioxide from pyruvic acid. And then we can add the electrons back from the NADH from glycolysis, and you form one of our favorite molecules, ethanol. Good old alcohol, right? Brewers or bakers' yeast actually do this process, right? Saccharomyces that we looked at the beginning of the semester will take glucose and break it down and form ethanol, right? Under certain conditions, it will do that. Another pathway they go is form formaldehyde and add a pyruvate to that and form alpha acetate, acetolactate, that one's a mouthful, an acid. And they can add protons and take away some carbon in the form of carbon dioxide and form acetonin. Acetonin then can be further reduced by adding electrons to it and form 2,3-butanol. Anybody know what test we did today? Tested for the presence and ability to do this fermentation pathway? This is the VP test. Very good. Right? So the VP test tests for 2,3-butanol, the ability of the organism to produce 2,3-butanol, but what actual chemical are our reagents reacting with? An intermediate, acetonin. Right. You are actually detecting the presence of acetonin today. This is a rate-limiting step, right? So we have quite a bit of acetonin, not so much of the 2,3-butanol. So it's pretty easy to test for the presence of the acetonin. So the Voges-Proskauer Test detects the intermediate acetonin, which accumulates in the media of our MRVP tubes, right? Those tubes were labeled that because they allowed us to run 2 tests. The methyl red and the Voges-Proskauer. So another choice when you have pyruvate is you can form lactic acid, which that happens in our bodies, right? Under anaerobic conditions, we don't have enough oxygen, because we're still going to break down glucose by glycolysis, and this electron carrier has got to be regenerated, so we form lactic acid. All right, the buildup of that can cause serious problems. Lots of microbes have this ability. Lactobacilli, part of the reason why they get that name, have the ability to produce this particular acid. And this is used in the bread making process. I think even for sourdough bread, because acids have a sour taste to them. And as I said, humans, right, we do this briefly. Another step, again, regenerating our electron carrier can form another acid, propionic acid. No organisms actually have evolved to go from lactate, though, to pyruvate that we're aware of. They do it a little bit backwards. They convert pyruvic acid into oxalatate, which remember is one of the intermediates in Krebs cycle. And then they convert it into an acid known as succinate. And then that acid, and again, notice these are similar steps to the Krebs cycle. They're using enzymes that are already there, right, but in a different way. And that's how they form propionate from succinate. So it's actually I can't even say this type of modified pyruvate that enters into the cycle. Peter's very detailed oriented. So pyruvic acid to acetyl-CoA. And then, again, right, hydrogen is picking up electrons here. Hydrogen protons picking up electrons. And then it can enter into the Krebs cycle, right? And again, pieces of it doesn't go the whole cycle, right? But it uses portions of the Krebs cycle to produce other acids and alcohols and ketones. So, there's a whole bunch of possibilities that can happen. And in this case, they don't even convert. They don't even take any electrons from it before they enter in. And in this case, they form another acid, formic acid. So they have lots of choices, and it depends on the organism and the environment in which it's in as to what it's going to do. What is it going to do to regenerate those electrons? So our phenol red broth tubes detect gas and acid production. They also allow us to detect deamination of proteins. The methyl red test detects mixed acid production. So when they produce a lot of acids that accumulate in these tubes. So possible acids that can be produced from fermentation. Formate, which is a 2-carbon, acetate, propionate, lactate, butate, succinate, right? Notice the 8, right? These are acids. Alcohols, notice they all end in OL, right? Methanol, ethanol, which is one we like. Isopropanol, I wouldn't suggest drinking that one. Butanol, something we usually burn, right? And a relative of butanol, the 2,3-butanol, right, that sometimes can be produced, and that's what our VP test tests for. Acetonin, not the same thing. Acetone, not the same as acetonin, which was the intermediate. This is a ketone. This is another acidic substance that some organisms will produce. And then you have good old gases, carbon dioxide or hydrogen gas. Hydrogen gas is unique to fermentation, although as we know, carbon dioxide is not, right? We produce carbon dioxide. So having produced carbon dioxide gas doesn't let you know necessarily that they're doing fermentation. Where if you could detect if it was hydrogen gas, you would know that that was produced from fermentation. Can you tell which gas is in our bubbles in our tubes? You can't tell. It's either hydrogen or carbon dioxide, or could even be both. We don't know, right? You'd have to do some research on your organism as to what it has the ability to produce. I know, for instance, E. coli produces both hydrogen gas and carbon dioxide gas. But again, the levels are going to vary depending on what type of environment the cell is in. So methanol can be produced by some anaerobes. And so they're actually not using oxygen as the final electron acceptor, right? They're using something else. This is toxic to aerobes and interferes with our electron transport chain, which is why you should not drink methanol, right? Or isopropanol, for that matter. And because it interferes with our, aerobic respiration, with our electron transport chain, it really, for humans, will affect the brain and the optic nerve, because those are constantly working, right, all the time. I don't know about when we're sleeping, but brain's definitely working. I don't know about the optic nerve so much. So these are questions straight from your homework, right? So which test detects this type of fermentation that we call mixed acid? That is our methyl red test, right? Or MR test. What reagent did we use? Methyl red. That's easy. The name of the test, right, and the reagent are the same. But what is methyl red? Methyl red is a pH indicator. It allowed us to detect by color changes, right, the pH. What do the products of this type of test of fermentation do to the pH of the media? Acids do what to pH? They decrease it, right? So it actually lowers the pH, right? Lowers the pH. Acids have a low pH. So as I said earlier, right, hydrogen ions are a hydrogen atom that's lost its electron. It's just a proton. And although they're smallest, they're probably the most important, right, to biological and chemical systems, right? The movement of these electrons and these protons, this atom we call hydrogen. It is so very simple, but so very essential. But also pH is the measure of this, of the level of this ion, right? How much hydrogen ion you have. pH actually stands for the partial pressure of hydrogen ions. So in the case of acidic substances, on the pH scale, it's an inverse relationship. They actually have a lot of hydrogen ions, protons, in solution when you have an acidic situation. The opposite is true when you're alkaline or basic. So chemists and biochemists pay special attention, right, when using the abbreviation pH. So pH scale, right, goes from 0 to 14, right? And so 7 is neutral. Below 7, like a pH of 4, is acidic. And notice that this is the concentration of hydrogen ions. Where in a basic situation, this is the concentration. And notice this is negative, so that's really small number, right? Negative 10 is really small number. Okay, so there's lots of different pH indicators out there. Some of the dyes that we even use are pH indicators, right? And so this chart shows you the different pH ranges, right, from 0 to 14, and where the different color changes happen. So ones that we utilize in this lab, we utilize Phenol Red. And so you'll notice that just below, right, doesn't have to be very acidic, just below 7, it turns yellow. Above that, it's going to be kind of an orangey red. And around 8, and you can't really see it well in this picture, it's kind of a pink magenta color. Remember we looked at those tubes in your racks you still have there? Right? That's when we've gone basic. So with Phenol Red, acid is yellow, basic is pink. In between is that orangey red color. Still with me? Okay. Methyl red in an acidic environment of 4.4, it's going to turn red. I mean, stay red. In the case, we started with it at an acidic level, right? So then when we added it to the media, if the media was basic, right, or not very acidic, because notice I said basic, but that's really in the acidic range. So we're really looking for very acidic. We're talking about around 4 for that methyl red test. A very acidic environment. And again, in between is kind of orange. The other one we use is in the citrate test. We use a pH indicator, Blumenthal blue. And it's blue in alkaline environments. Again, I don't really kind of show it here. But at 7.6 and up, it's going to be blue. So you'll see even crystal violet, right, in acidic conditions, changes colors. So, which test is for fermentation that produces 2,3-butanol? That's our Voges-Proskauer, our VP test. What reagents? We used 2, did we not? We used Buret's reagent A and B. But what chemically they are alpha-nethaphenol and potassium hydroxide. What is the intermediate product that we're actually detecting by using these chemicals? Acetonin. Not to be confused with acetone. These are different. Alright, so Glycolysis, right? We can break down glucose, produce pyruvate. We can convert it into other acids. We can produce acetonin, which then can be converted into the 2,3-butanol. When we run this test, though, the alpha-dithaptanol, the first reagent, binds with the acetone. And then when we add the second reagent, and this is why I told you guys to flick it a lot, because we need to add oxygen for this reaction to go forward. Right? So that's why by flicking it, right, we're adding oxygen from the environment into the tube. Diacetyl forms, and then guanine, which is a peptone already found in the media, produces the red color that we saw. So, methyl red test, which one's positive? The red one, right? And again, it's that really low 4.4 pH or lower that's going to give us that nice red color. Above that is yellow or orange, which is negative. Oh, that's all right. We'll put them up. Red. The answer is red for both these tests, right? Although these pictures, for some reason that semester, it was kind of a pinkish color, but it's still a shade of red, right? So you can see there's a nice strong positives, right? Sometimes you'll get a little weak positive. If it's any other color than reddish, it's negative. So some of you guys may have gotten like a little amber color at the top. That would have been negative. And again, that's because of a chemical reaction. So for each of these tests, each group, right, and we actually just did it as a class, we ran positive and negative controls. And those thankfully came out as expected. Why do we do that, though? What happens if my positive control did not come out positive? Can we trust your results? No. If my negative came out positive, woo, that's bad, right? That would invalidate all our unknown tests. So what could have went wrong, other than me screwing up, which I slightly did today? Contamination. What about the reagents, right? What if somebody put the wrong reagents in the wrong bottles, or what if they're expired, right? So we want to make sure that we have reliable results. So we run controls, in this case a negative and positive, so we know what they look like, too, for comparison. But also to make sure that our reagents and everything is working properly. This is our procedure that we followed today. Right, we split it up into 2 tubes so we could run each test separately. Right, and we really only had to wait about 10 minutes. Right, but you could potentially have to wait longer. Why would it take longer sometimes? When we did the methyl red test, we read it right then, right? Because what is methyl red? It's just a pH indicator, right? That happens within seconds. What are we waiting for to happen when we run the Voges-Proskauer test? What do you refer to this as? What is that? The chemical reaction, is it not? Do those sometimes take time? Yeah. And you got to get enough of this red color produced that you can actually see it. So chemical reactions take time. So false negative result for the VP test. Poor sensitivity or poor specificity of our test. Sensitivity. And again, because you got to get enough of the red color produced that you can actually see it. It's a pretty specific test. Right? It's a sensitivity issue. Do you have enough of the acetonin to convert enough of the reagents into the red color that you can visualize it? So I had to wait, right? Why'd I go backwards? I had it here. Alright, so the acetone is quickly converted into the end product. There will be very little amounts of the intermediate product left to detect. But that's not the case, right? There are other circumstances when the concentration of acetone might be low. For example, perhaps acetone lingers around for a long time in a particular species of bacteria, but not much acetone is generated in the first place due to a low inoculum, right? If you didn't get enough bacteria in there, didn't get enough growth, weren't inoculated at the right temperatures, right? So there's several variables that could affect this test, right, that would necessarily give you negative results. The false positives we saw today, I would probably more likely contribute to possible contamination. So, why were you told to shake the VP tubes after reagents were added? We needed to add the oxygen, right? We need the oxygen for this chemical reaction. So for the methyl red test, it says 2 drops. 2 or 3, doesn't matter. It's just a pH indicator. Just need enough that we see a color change. Alright, red right away, right? Because as I said, it's just a pH indicator. It's just a pH test. It happens within seconds, the color change. If positive, it'll be red. If negative, right, the red will mostly disappear, right? You'll see the kind of yellowish color or an orange. Both are negative. It's only red that's positive. And again, that'll be that low 4.4 or lower pH. So for glucose, right, some organisms can produce a mixture of acids. So E. coli, I think is the example they have here. Yep. So E. coli, when given glucose, it can generate lactic acid, carbon dioxide, hydrogen gas, ethanol, acetate, formate, and succinate. There's a whole lot of acid and some gas in there too. Right? So we're not testing necessarily for individual acids, but the fact that it produces a lot of acids, right? So hence the name of the test, mixed acid fermentation, where you're getting a mixture of several different acids. So why is the methyl red test ready immediately, and the Voges-Proskauer can only, it's suggested that you wait at least 60 minutes? What's the difference? Methyl red test is just pH. Voges-Proskauer, what has to happen? Chemical reaction. Typically, we would incubate this for about 2 days. But you can go longer. And I think before we tested these today, they've been incubated for like a week, right? Hence why, if some of you guys had minor contamination, why you might have got false positives. Right? Probably been less likely if we had tested it earlier. But why would you let it incubate a long time? What happens over time? Either test, right? So if we're giving it glucose, what are they doing with it? They're fermenting it, right? So in the case of like E. coli, it's producing all those acids. The longer it grows, the more what's going to be produced? Acid. And same thing with the acetone, right? The longer you incubate it, the more it's going to be produced. So therefore, the more likely you're going to get a positive result if it truly is positive. You're not going to have to deal with so much your false negatives. So if you're your methyl red test came out negative and was supposed to be positive, right? We could have incubated a little bit longer. Considering how long you guys have been incubating, those of you guys that were supposed to get positive results did. More than enough time to produce lots of acids. So why do we do these things, right? Well, we want to know, can they do mixed acid fermentation? Can they do 2,3-butanol fermentation? And what is butanol? Is it an acid, a gas, or an alcohol? It's an alcohol. It's an alcohol. Alright? This is actually the only test we run that detects the presence of an alcohol, or whether they can produce an alcohol. How do you perform the test, right? Make sure you perform it accurately, and then record your results, what you guys are doing. What do these things tell us about our organisms, right? If they're positive for the methyl red test, it means that they can ferment the glucose and produce a lot of acid. Right? If it was negative, they're not doing mixed acid fermentation. VP test. If it came out positive, you know your organism is converting glucose into acetonin, which can later be converted into 2,3-butanol and alcohol. If your organism didn't do that, right, it was negative for the test. Phenol red. These are my favorite ones. They're pretty, huh? So we have phenol red, which is a pH indicator, right? So we can detect by color change what pH we're dealing with. And then we also have a special little tube that's upside down in yours to trap any gas production. We call this the Durham tube. And so we get a variety of different colors based on the different levels of acid or basic products. And we use a coded system when we're coding these tubes. A for acid, and we know it's acid because it turns yellow. We code G for gas, right? And we know that because you will see a bubble or large bubbles in your Durham tube. If you don't see any bubbles, then we code that as negative for gas. This is our negative control. This is a tube that looks pretty much the same as the negative control, right? No acid production, no gas production. You would code that minus minus. The problem with this is it like this because your organism didn't grow? Or does your organism truly not ferment or utilize the protein source? This is rare. We didn't have any of these, right? Everybody was yellow. A few of you guys were supposed to be K. We're waiting on it. Actually, might stick yours in the incubator a little bit longer, see if it'll cooperate. K means that K actually stands for alkaline, but we can't use A and A, right? So we go to the next defining letter, and K represents a basic or alkaline environment. In this case, they didn't do fermentation. Instead, they use the protein as a food source, and when they do that, they produce basic byproducts and are going to turn the media this pretty pink magenta color. So this is the code that you guys need to learn what it means, right, for each one of these tubes. Now, we did glucose, right? But from the manufacturer, you can test for tons of different sugar or sugar alcohols, right? So different ones to see if they're able to utilize these sugars. And then the base broth itself doesn't contain any sugar at all, just a protein source. And we're going to talk about that. So this is the formula of what we utilize, the food that's in the phenol red. So we have an amino acid source. This is casein. It's a protein from milk, actually. It's actually what makes milk white. It's animal proteins. And it's partially digested with pancreatic enzymes. So basically we're giving them baby food, pre-digested food, so they'll grow fast and be happy. We have sodium chloride to have an osmotic balance, right? We want to keep a good osmolarity to the media. Glucose, as I said, is our sugar source, our carbohydrate they're going to use for energy and electrons. Phenol red, of course, is there as our pH indicator. And then water. And balance the pH to around normal, right? So around neutral. So they really only have two food choices in these tubes. They eat the protein or they eat the sugar. That's it. So what are some of the common end products of pyruvate fermentation? So what did we say? Acids, alcohols, one more, gas, right? So gas is carbon dioxide or hydrogen gas. Alcohols, there's a whole list of them, right? An even longer list of acids. Those are the general categories of general products that organisms are going to produce when they ferment. Which one of the three cannot be detected in the phenol red test? Can we detect gas? Phenol red test. Let me rewind. Can we detect gas? What's that bubble? Yeah. Yes. Can we detect acids? Yeah, that's with a color change, right? To yellow. Can we detect alcohols? No. We already said what test do we run is the only test that we can detect in alcohol. Which test allows us to test for 2,3-butanol? BP. Okay. So what's the pH indicator in this media? It's the name of this media. It's what? Phenol red. What color does the media turn if acid products are generated? Yellow. Right? So notice it's not a very low pH, right? Just below neutral. Around 6. That you'll get yellow color. What color does the media turn if alkaline products are generated? It's that pink or magenta color. Peter, I think, is not happy with my picture. It's not pink enough for him. He wrote a question mark down here. He's like, I don't think it really looks different than this, so I guess I got to get a better picture. Okay. What does the derm tube help detect? Gas, right? By the presence of that bubble. If glucose is fermented, what happens to the pH of the media? Well, what's a product of fermentation that would affect pH? Acid, right? So the pH goes down, right? Lowers the pH because we're producing acids. And we're going to get what color? Yellow. Peptone, right, as we saw in the formula, is a mixture of proteins and polypeptides. Some organisms can use that as a carbon and energy source. And a process referred to as deaminated. In this process, it produces ammonia. Ammonia does what to the pH of the media? It's basic, so it increases the pH, right? It's a basic compound, right? It actually sucks up hydrogen ions. And so therefore, right, hydrogen ions decrease the pH, right? Make it acidic. Ammonia increases. So, increased. Pink. When conducting this test, some microbiologists, not us, include two sets of broth. One with a glucose that contains just the base broth, no sugar, and one that contains the sugar. We also do an uninoculated control, right, to help with the reliability of the test. Those controls I have in the racks for you guys. What do the bacteria have to eat in each tube? And what is produced when the bacteria break down when they break down proteins? So, our tubes all had a choice, right? Glucose or protein. Sometimes they'll give it to them with just the protein, no glucose at all. So, if you have the choice, what will happen if they eat the glucose? What will we see? They're going to ferment it, aren't they? Right? So, the pH is going to go down, and we're going to get that yellow color from acid production. If they eat the protein, what gets produced? Ammonia, right? Which is basic or alkaline, and the pH is going to go up, and it's going to turn pink. Ooh, this is a tricky one. What happens if the bacteria eat all the sugar, then begin to eat the protein? So sugar is going to turn yellow, and then when they start eating the protein, it's going to start turning orange, right? Because that's in between to pink. And it really depends on where the final pH is, right, as to what color it'll be. But, you know, sometimes, and actually, if she didn't shake her tube, is it Chanel that sits right behind you? Chanel? I remember? Yay. Chanel, yours actually might have had a little orange layer on it, did it? No? Okay. Your Phenol Red tube. And yours is one that eats the sugar really fast. It's one of the reasons why I take them out of the incubator and I refrigerated them for you guys. To keep them from eating up all the sugar and then start eating the protein. And so yours does that even within the 48-hour time frame. So sometimes you'll start seeing a little bit of orange because it's exhausted the sugar and it starts eating the protein. Protein only, the only possible result that could happen is for it to turn pink. Right? There's no way you can get yellow when you do not have any sugar. Everyone understand that? So in this table, they're talking about comparing these two tubes, right? And that set of tubes is like a control set. This will also help you guys learn your code. Right? So AG stands for acid, right, and gas production from fermentation, which means they ate which food source? The sugar. Now, notice this is phenol red with glucose. That's a possibility, right? We have, what, 4 different possibilities, 5 possibilities that could happen. But if you have just the base broth, the tubes only contain protein. If someone coded that AG, that means they're seeing a yellow tube with a gas bubble, that you had fermentation and gas production. How the heck did that happen for protein? Didn't. Somebody screwed up, right? Somebody labeled a tube wrong. Something went wrong. You would not trust the results from that test, right? They would not be reliable. No way. Now, again, this is possible. Organisms ferment the glucose, we get acid and gas because glucose is there. In our protein-only tube, we see no fermentation. It's coded -, which means, what color is that tube? What color is our uninoculated tubes? Kind of an orange color, right? And no gas bubble. So, is that a possibility? Could it do nothing when we just give it protein? Now, given the choice, this organism is like me, right? When given the choice between sugar and protein, it chose the sugar, right? He's like, oh yeah, sugar and gas production, right? Yellow tube with a bubble. But when giving only protein, it degraded the protein. We label this K. What color is that tube? Pink. Is this a reliable result? Sure. Right? They were given a choice here. They chose sugar first. As do I. Many times when I'm cooking the hamburger at home, I'm eating some candy out of the fridge while the hamburger is cooking because I'm hungry and I don't want to wait. Not the best choice nutritionally, but microorganisms are like me, right? They want that quick, fast energy usually. And so they're going to pick the sugar over the protein most times, if given the choice. But notice here, no choice, right? So they're like, okay, fine, protein, I'll eat it. Twist my arm. So you guys see how that would be a reliable result? Okay, so for the next one, again, given the choice, acid and gas, right? Yellow tube with a bubble. What does this mean if we code it A minus? Acid, no gas. What color is the tube? Yellow. Okay, how the heck did that happen if there's only protein in that tube? Somebody messed up, right? You can't get acid production without sugars. This is not a reliable. Right? No, what happened? Alright, K means alkaline end products from the degradation of protein. What color will that tube be? Pink. Given the choice, what did they choose? Sugar or protein? Protein. So when we gave them just protein, of course they ate it again. So is this a reliable result? Sure. Next one. Chooses the protein again. Smart microbe. In the protein only tube, if it's minus minus, it's an orange tube and no gas, right? What happened here? It should have ate the protein again, right? Why didn't it? Something went wrong. This is not reliable. Should have done the same thing, right? Here they were given the choice and they took the protein. Here they were given only protein. They should have ate it. Why didn't they? And an additional question would be, did the organism even grow in this tube? So A minus, remember, is what color tube? Yellow. Did it produce any gas, though? Nope. Alright, so it still ate what? If we have acid, it ate the sugar, right? But then when given the choice, right, it chose sugar, but over here, just protein, it went ahead and ate the protein. Is this reliable? Sure. Again, just like before, right, given the choice, they prefer sugar, but they will utilize the protein if only the protein is available. So fermentation, yellow tube, no gas bubble, and our protein only tube, if you code it AG, it's what color? Yellow. And what does the G mean? Gas. Where the heck did the acid and gas come from? This is a protein only tube. Somebody messed up, right? And not to mention, look at over here, no gas at all, and then all of a sudden you have gas? Where did that come from? Right? That doesn't happen. That wouldn't be reliable. Alright, suppose you inoculate base broth with a slow-growing fermenter, Julia's organism. is a slowpoke, right? And I know this about this organism, but if you didn't know, right, and you get turbidity, right? You see cloudiness, so the organism is growing, but it's still kind of an orangey color. Is this a false positive or false negative? This is a false negative, right? Because we expect it to be positive. And actually, hers isn't even a fermenter. Hers has a deaminator, but still slow at that too. So why would we have this problem? Because we got to have enough of the acid produced that it changes the pH, right? So is this a specificity or sensitivity issue? Sensitivity. Everyone understand why it's sensitivity? Right? Again, we have to have enough of something. And in this test, it really doesn't take much. Early formulations of this media contained a small amount of carbohydrate and occasionally produced false alkaline pink results after 48 hours. Why do you think this happened? So if they have a little bit of sugar, what happened? They ate it, and then they started eating the protein. So how do we fix this problem? Add more sugar, right? Or what else could we do? How long do we incubate them for? 48 hours. If we shorten the incubation time, will we catch them still eating the sugar? Yeah. But they, of course, increase the amount of sugar. Right? So add more sugar, or you can incubate for a shorter time. So especially if I had left Chanel's organism in the incubator, it would have turned pink, and that would have been an accurate result for hers. Because in truth, it picks the sugar first. Don't do that to us. It's just a blip in the... Huh?
There's supposed to be a storm coming through.
Yeah. So, let me, So for Phenol Red, we actually are kind of asking two questions, right? Can it ferment the glucose? And if it does it produce gas? Or does it utilize the protein instead? So we just inoculated our tubes, we incubated them, right? And then we looked at our results and we coded them based on what we saw. And this tells us something about our organisms, right? What is the organism is capable of doing or not, right? So if we had yellow tubes, we knew it ate the sugar, it fermented the sugar, it produced acids which lowered the pH and turned it yellow.
Oh, yeah.
Right? And if they produced gas, the gas was trapped in the Durham tube, right? And so we know that it fermented the sugar. So these types of descriptions are what I expect you to write on your pages, right? Tell me specifically about your organism. So the organism, if it turned pink, is capable of deaminating the proteins. It didn't ferment the sugars. You got an increase in pH, right? Because of that ammonia being produced. And you get the pink color. That's a complete description of what happened for your organism, right? So again, for these tests, I want to know that you know, right, what happened, right? How you know what happened for your organism, right? You know if it's a yellow tube, it fermented the sugar, it produced acids which lower the pH and turn it yellow. If it produced gas, you know that because you see a bubble that trapped gas in your Durham tube. right? So you say, I know mine produced gas, right, because there's the presence of the bubble in the derm tube. If yours had turned pink, right, you know your organism deaminated the proteins, produced alkaline product of ammonia, which increases the pH and turns it what? Pink. Still with me? All right. Moving on to citrate. So the citrate test is actually for the test, is testing for the presence of a protein embedded in the membrane called citrate permease. And what permeases are membrane-bound proteins, and they help facilitate the movement of molecules across the membrane. So in the case of citrate permease, it moves citrate from outside the cell to inside the cell. I couldn't find a citrate permease picture, so I've got a sucrose transporter here. Same principle though, right? It is going to help transport that sugar into the cell. So what's the only carbon source in the media for that organism? Every organism needs a carbon source. It's the citrate. The sodium citrate, only carbon source. That's it. What's the only nitrogen source? Ammonium dihydrogen phosphate. Since this media only has one carbon and one nitrogen source, what will happen to these organisms if they can't use these molecules, if they can't bring them into their cells? They'll die. They'll die. So those of you guys that had green tubes, you probably didn't see any growth, and if you saw anything, it might have been a heavy inoculum that you put on there originally. Those of you guys that had a blue tube, you probably saw quite a bit of growth on that slant. So again, we have a pH change that happens because when they use the ammonium ammonium dihydrogen phosphate, when they use that, they're going to produce alkaline substances. So in this case, right, it's going to go basic range, so it turns that really pretty blue. Where the media starts out, right, more of a neutral range, a little acidic even, in that green color range. So what color does the media turn if the organisms are utilizing the only nitrogen source? It turns blue, right? pH increases. If the media is green and you see growth of the organism in the tube, is this a positive result for the utilization of citrate? If you see true growth, yeah, because the only way it can grow, right, is if it's utilizing the citrate and the nitrogen source. So if it's truly growing, it is a positive result. For you guys, again, I let these go for a long time so that we would build up plenty of the ammonia, we'd get plenty of growth, and there wouldn't be any question on whether it was positive. And definitely, if you didn't get any blue change by then, your organism died. And hopefully, it wasn't dead to begin with. It just died because of these conditions, right? And you ended up with a negative result. The problem that some people run into sometimes is, like I said, they confuse a heavy inoculum having put a lot of bacteria on it to begin with as growth having happened, right? But you would have substantial growth if your organism really is utilizing. The problem is, right, again, we're dealing with a color change, right? So if a citrate positive organism might not produce a color change just yet, right? Is this a false negative result? Yeah, if you don't notice that there's growth, right, and you don't incubate it longer, say, because you don't have enough. So again, is this going to be poor sensitivity or specificity? We need enough ammonia to create a color change in the pH indicator. Sensitivity. Poor sensitivity. So again, for this test, we want to know, does your organism have citrate permease? Can it bring citrate in? And then once it brings the citrate in to use as a carbon source, it's going to utilize the ammonia, it's going to produce the base, and then you're going to get this blue color. What happened to your poor organisms if they don't have citrate permease? They died. And you got no color change, right? So the organism either is or is not capable of fermenting citrate and consuming the ammonia as their sole carbon and nitrogen sources. Last test, catalase. This is one of my favorite ones, though. So, flavoprotein is an electron carrier that passes electrons to oxygen. And this is one of the ones that screws up sometimes and is going to produce those toxic forms of oxygen. So when that happens, we need enzymes that are going to destroy those toxic forms of oxygen that we saw earlier. So super deoxide demutase is the enzyme that will convert that superoxide radical, that highly charged oxygen, into safe oxygen and hydrogen peroxide, which is another thing that can sometimes be formed from the electron transport chain, right? So we have another enzyme that can now take hydrogen peroxide, which is still quite damaging, just not quite as damaging as the superoxide, convert it into water and gaseous oxygen, and that's why we get the bubbling, right? Notice I said aerobes. So these are ones that are living in the presence of oxygen, they're utilizing oxygen, they're utilizing that electron transport chain, right? So they're going to produce superoxide radicals sometimes and hydrogen peroxide. And you have to have some way of detoxifying these substances and making it safe again. So how we tested for this is we tested first on a slide, right? You added your organism, then we added some hydrogen peroxide to it. Most of you guys got bubbling. Sometimes it's kind of hard to see on the slide, again, depending on how many bacteria you have on the slide and how many of them are actually still living. So to back that up, we went ahead and did the test also on the slant, where you have a large number of bacteria, right, and usually get a really good result. So would a false positive result from the reaction between the inoculating loop and the hydrogen peroxide be caused by poor specificity or sensitivity of this test? This is the one problem where you get a sensitivity issue. Specificity issue, right? You don't know if it's the metal loop, right, or your organism that's causing the bubbling. And that's why we always add the bacteria first and then the hydrogen peroxide. Today it wouldn't have really mattered because I cheated, so we didn't have to drag out the Bunsen burners for one thing. We just used the plastic loops. If you have a weak catalase positive organism, right, so on your slide, right, you don't really see your bubbles. One of the ways that would help you see your bubbles if you looked at it under the microscope, right? So what if you had trouble seeing it, right? So you think it's a negative result, and you record it as negative. Is this a problem of poor specificity or sensitivity in this instance? This is a sensitivity issue, right? You can't really see the bubbles. Why is it advised to perform this test on a known catalase positive organism along with the organism you're checking? What's the reagent we're using? Hydrogen peroxide. Can you tell hydrogen peroxide from water by looking at it? No siree. So you need to make sure that your reagent is good. Right? So you always have your positive control. It's also why we keep it in a dark bottle, because light can cause this reaction. Over time it'll turn into water and the gaseous oxygen that will be released. You shouldn't. I wouldn't suggest it. Because there would still be a small amount, but not a significant amount of hydrogen peroxide. The problem is it'll be mostly water and not hydrogen peroxide, and therefore not do what you want it to do. Now, as I said earlier, they suggest that you no longer use it for wounds. I want to make sure, is this my last one? No, we have one more. And so why was it that I put the hydrogen peroxide on the uninoculated tube? That was another control, right? To make sure the media doesn't cause bubbles. Because if we do it with the organism in the media and the media causes bubbles, you don't know if it's the organism or the media that's causing the bubbling, right? We have to have that control. So we're seeing if that exhibits the catalase activity. And remember, this is kind of linked to organisms that respire aerobically, right? And so this brings to the question of our FTM tubes, right? Remember those? Most of your organisms grew aerobically, right? Some of them even grew anaerobically, right? They grew at the bottom of your tube. So definitely, the fact that all of them grew under aerobic conditions, probably most of them were utilizing oxygen, yes? Definitely your strict aerobes, which stayed right at the top of your tube. Other ones were probably what classification? Facultative. Kate, what do you think about yours, though? That it was growing anaerobically? I agree. But it doesn't have catalase, right? So it's probably not growing aerobically, but it grew all throughout your tube, didn't it? So which one was it probably? Look at your list. It's on the list.
Air tolerant.
There you go. So it can tolerate, but it doesn't have catalase, right? But what other enzyme does it probably have? The superoxide demutase. That's probably what it allows to tolerate. And it doesn't really need catalase because it's probably not generating a lot of toxic oxygen products because it's not using oxygen, right? Unlike the rest of your guys' organisms, we're definitely using oxygen, right? So they definitely needed to have catalase to protect them. So there's an old wives' tale out there where if you put hydrogen peroxide on a wound and it doesn't bubble, it means it's not infected. And okay? You are catalase positive, people. You're aerobes, right? You have catalase. If you damage your cells, they're going to cause it to bubble. So if you pour it on you, it doesn't bubble, it means you just poured what on you mostly? Water. It's old hydrogen peroxide, right? Pitch it and get a new bottle. Or better yet, don't use it on your skin anymore anyways. Use it to clean clothes. Use it, to rinse your mouth if you really feel the need. That's really gross, by the way. I had to do it once. I had my wisdom teeth removed before they came up with these special mouthwashes for that. I ended up with dry socket. It's not cool, right? So, but I spray my retrainer with hydrogen peroxide, right? I don't put it in my mouth. I rinse it off, right? Clean my earrings with hydrogen peroxide, right? Again, that bubbling action of any bacteria and stuff that's on it. But you certainly don't want to ingest it, right? And that's why it should only be used topically. And as I said, they suggest now that you don't, because it damages even your cells, right? And that bubbling, right, action really helps break things up. So it's a good cleaning agent. Just don't put it in or on your body, right? And as I said, your catalase positive should always bubble. Always, always bubble if you were to put it on you. Okay? The only reason it wouldn't is if it was old and then you had mostly water. Make sense? Alright, so at this point, as I said, you should pretty much know which organism you have. If you're dying to know if you're right, you can ask me before you leave.